All good to go, so thank you. So the last real information dump from me to you is on organ on a chip. And while none of you are biologists, at least half of the research of what's happening in microfluidics at the moment is working with all kinds of cell and organ on a chip systems. So I thought if we are talking about microfluidics in 2 weeks, I can't possibly course and not even on the touch of like about half of what we're doing. So an organ on a chip device, also referred to as OOC, emulates the physiological environment and functionality of human but also animal organs on a microfluidic device. So this is the way that can be graphically represented. We can look at having a little chamber where we have some heart muscles. And if heart muscles are happy, they actually start contracting by themselves, and they do that in a synchronized manner. So that's really nice. If you've got a good cell culture system ready for heart cells, and then the muscle fiber cells, they nicely align. And the beating, the contraction motion, you actually don't need to have a whole part of heart tissue in order to get that contractility working. Another tissue that is functionally important for looking at pharmacology as well as understanding disease pathways is the bone. Not necessarily because the bone is strong and holds us in our shape, but the bone is also very important in our immune function. So if we have a microfluidic mimic of the bone, it also means that we've got a mimic of how the immune system works. We can create microfluidic alternatives to the lung, where we can look at the absorption of oxygen and the release of CO2, so the exchange of air. We can look at the kidneys, which are really important if you start looking at pharmacological models, because the kidneys are our organ that are important for the excretion. The liver, of course, is very important for our metabolism. Where is he sitting? There we've got a little liver chip. And then we've got the gut, which is very important to study the absorption of drugs and nutrients to the body to get into our circulation. So here you can see that by getting a fluidic network where we have cell culture systems of a couple of different organs, we can actually mimic a lot of the body's functionality. It takes a long time to develop all of this because all of these cells require a slightly different microenvironment, both physically, so some cells like some structures, other cells like others. And cells are very finicky that they like certain chemistries, and the chemistry in our body is not homogeneous. So we will need to adjust the cell culturing conditions depending on the cell types and the organs we're working with. So we've got the ability to cell on a chip. In parallel, we've got in the pharmaceutical industry the discovery pipeline happening for new drugs, where it starts off with basic research where pharmaceutical chemists just make whole libraries of compounds and do either simple enzyme-based studies or whole cell studies to screen the drugs to find if a drug is effective. After that, a lead is selected and optimized. Once all the in vitro assays are promising, then we move on to the preclinical research where we do animal studies. After there have not been too many rats or mice being killed, we move on to to human studies, where we start off in phase 1 in small groups, and then increasingly go into large groups, moving to phase 2 and phase 3 trials. After all those tests has been done, reports are written from anything happening in the basic research all the way to the phase 3 trials. All those documents are being considered by legislative companies. Then, if everything is found properly, it is registered by the FDA or any national equivalent, and then the drug can go for manufacturing and be sold on the market. That is so we have the drug discovery pipeline, where we have some uncertainties around the toxicity and efficacy of new drugs, because we humans were not mice, and our pharmacology, our distribution, it is actually not really well mimicked in mice. So in the pharmaceutical industry, we've got kind of a disconnect between our preclinical and our clinical studies. So having a closer model to a human than a mouse would actually really benefit this pipeline, because we would be much easier to translate from our preclinical to our clinical range in drug discovery research. At the same time, from cell culture, it is well known that cells are not happy in a petri dish or on a flat substrate. Cells in our body are always sitting on spherical, irregular places. We are not flat creatures, we are three-dimensional. So in these cell cultures, we've got a couple of problems. If we're sitting on the bottom of a petri dish, we can't absorb any nutrients from the bottom. It means that one part of our being as a cell is flat, whereas the other part is still bulging out, so we're no longer symmetrical. Spreading and migration becomes hard. Adhesion can only happen in the X plane, because as a cell, it is impossible for me to reach the ceiling of this flask. And the substrate is really stiff, whereas nothing in our body is as stiff other than maybe the bone as a culture plate. So in the history of cell culture, there's been a growing awareness that cells are actually much happier in 3D environments that truly mimic the environment that they are present in a cell, which means that we have different nutrients that are available, either homogeneously or heterogeneously from our complete environment. We don't have any prescribed polarities. The cells here can functionalize and differentiate how they want. There is a matrix holding it all together with the fibrils. And adhesion and distribution can be managed and organized across all three dimensions. The last advantage of the 3D cell cultures that typically use hydrogels or other soft materials as a support is that the low stiffness of the substrates make a much more bio-relevant environment, which means that we can expect the cells to also behave in a much more bio-relevant manner. So we have a level of dissatisfaction in the pharmaceutical industry about the use of animal models and the relevance of them. And at the same time, we've got a societal pressure to decrease the animal testing in the pharmaceutical industry. If we combine with the realization that 2D cell culture is not biorealistic, but microfluidics provides the opportunity to fluidically control the 3D environment at the similar dimensions as the cell take place, it makes sense that we start conducting these cell-based studies in created microenvironments and use microfluidic principles in order to design these devices and create the administration of nutrients and the transport away of waste products in cell culture. So a lot of different developments have happened, and if we look in here over time, in the late 80s we started with some microfluidic fabrication and the creation of microchannels, and with that came increasing capability that is really useful for cell culture. We looked earlier this morning at a concentration gradient to look at the different spores that were developing. By making gradients, you can screen different drug concentrations on cells without having to make different dilutions. A lot of work has been done on perfusing cell culture to add new nutrients and remove waste products. With that happens a lot of understanding of the flow and the physics, and understanding which cell type is happy with what flow rate, because the flow rate indicates the shear stresses on the cells. And for example, blood cells have very little problem with shear stresses because blood cells are always present in a flowing environment, whereas liver cells hate shear stress because they don't like flow. A lot of different methods have been developed to make so-called spheroids, which is not a single cell, but actually a cluster of cells together. We have been able to do single cell studies looking at mechanical and material cues to see if cells, particularly stem cells, can differentiate any difference. And there's a lot of learning that has been done in culturing the cells and see how we could mimic the pharmacological assays that we would normally conduct on either 2D cell culture or in mice, how we could better do these using human cell lines in microfluidic devices. So the degree of the complexity over the past 30, 40 years has significantly increased from being able to just make a chip with a couple of channels to having completely absorption, distribution, metabolism and excretion studies of the drug, so being able to do complete toxicity screens using microfabricated devices. In cell culture itself, a lot has developed as well. And not only have we moved from the flat Petri dishes to working with the more spheroid-like cultures, he can see that in the past 10 years, creating different organ-like structures of one, but preferably multiple cell types, that self-assemble into a functional unit that doesn't necessarily have the shape or the size of our livers in our bodies, but that do have the different cell types supporting each other to conduct the function of the liver. That has been the big findings in cell culture, and here you can see a couple of examples of different types of organoids with heterogeneous cell cultures that have been combined into little clumps to have human-like behaviors. Here we can have that a little bit more zoomed out, and you can see that we've got differentiated liver organoids being formed at around 2012, cerebral organoids that mimic the functioning of the blood-brain barrier. We've got kidney organoids. Lungs were a little bit more temperamental, but here we've got some lung organoids where we can also have a liquid-air barrier because the lungs have an interface between air that we inhale and the bloodstream on the other hand. And we more recently we have organoid models of the blood vessels to look at their contractility and vascular pathologies. And as you know, vascular disease is a big problem in the humans as well. And there's little organoids that have heart cells created from pluripotent stem cells that actually contract all synchronized because in their little functional units they're talking together. So I've been talking about cells, spheroids, organoids. They all are different steps of complexity. So we started off with 2D cell culture where we just had an isolated cancer cell line. We throw them in a Petri dish. If they're adherent cells, they sit on the bottom. If they're suspended cells, they sit somewhere float in the solution. But they're all single cells that don't communicate together and are not connected. If we go one step up from them, we get these cells to organize into a cluster that we call a spheroid. And spheroids could be comprised of one cell type, but also consist of multiple cells. So if we talk about the lung, we've got epithelial cells that are the interface with the air and the liquid, but then we also have got the endothelial cells and a whole range of functional cells sitting underneath. So in a spheroid, we can have these cells living next to each other and interacting. A step up in complexity then is when we've got the organoids. And the organoids are a step up because we also start having the functionality of these cells that they would have in a human organism. So we have multiple cell types. We have a scaffold sitting on the outside. We have tissue-like characteristics. So if it's a liver organoid and we add a drug, we get metabolites being formed. And the nice thing about organoids is they actually have an extended lifespan. So we can do studies over days to weeks at a time, which means, again, we are a lot closer to mimicking or getting the benefits from both we know from the mouse model, but the mouse was not biorelevant, but we can't quite do some of the early stage drug testing on humans yet. So organoids provide a really nice platform to do that. One step up that we won't be touching is the assembloids, and they are the parts that are used then later on for 3D printing. Considering we only look at microfluidics here, we draw the line at the organoid level. So cells can come from a whole range of different sources. And if you read some of these papers, they were always indicating where they got the cells from. What is better or not is probably dependent on what's available in the lab and the kind of studies that you're doing. But you can start with pluripotent stem cells. There are cells that haven't been programmed yet, and based on the chemical cues that you give them, you can then get them to differentiate as a neuron or a gut epithelial cell. So they are a source of introducing functional cells into a spheroid. We can actually take primary cells from the original tissue. So we can get a biopsy from a tumor and create spheroids that actually contains a patient's own cancer cells. which is a really nice platform if you want to do drug screening, because if you want to know if a drug is going to be effective on a specific patient, what better model to use than the particular cancer that this patient is suffering from. Additionally, we can buy a whole range of different stable cell lines or genetically modified cell lines from commercial sources. You can have them in the freezer, work them up, and grow organoids out of them as well. So there's different approaches that you can use to start making the organoids. Then we've got a wide range of different designs and structures that we can use to study cells and organs in microfluidic systems. So all of the ones on this page will be going through one for one, starting with a mechanically actuatable co-culture device. So here we've got the microfluidic device where we've got epithelial and endothelial cells seeded across two sides of a membrane. In this case, if this is lung cells, we've got air flowing through our top channel, whereas we've got a liquid mimicking the blood or the interstitial fluid in the lungs flowing underneath. It's a mechanically actuatable device because we've got a PDMS device, which was a flexible substrate. If we apply vacuum to these microchannels, they will pull the membrane apart, and that causes the flexing that our lung cells experience when we breathe in and breathe out. Our lungs are not used to sit on a stationary surface. Our lungs, we continuously breathe, so every time we breathe in, all the cells are being stretched. Every time we breathe out, the cells actually relax again. So these devices can mimic that in a biorealistic way. Here we've got a little movie demonstrating that, and I hope that my internet connection will allow us to play this. There we go.
The lung on a chip is a microdevice that has the potential to replace animals for the testing of drugs and toxins. The device, which is lined by human cells, mimics the mechanical and chemical functions of a living, breathing lung. With every breath, air enters the lungs, filling and expanding microscopic air sacs. Oxygen is transferred across lung and capillary cells and into the bloodstream. This is also where aerosol drugs are absorbed and where infections and tumors form in the lung. The lung on a chip is crystal clear, flexible, and about the size of a small computer memory stick. But it contains tiny hollow channels created using microchip fabrication techniques. A porous, flexible membrane separates the two channels at the center of the device. The opposite sides of the membrane are lined by human lung and capillary blood vessel cells. This mimics the arrangement of lung and blood vessel cells in the air sac of the lung. Application of cyclic suction in side channels makes the entire flexible sheet and cells stretch and relax rhythmically, just like our lung cells do when we breathe. In the lung on a chip device, air flows over the top of the human lung cells, and a liquid medium containing human white blood cells flows below the capillary cell layer. To test how well the lung on a chip device replicates the natural responses of living lungs, we introduced bacteria into the air channel to mimic an infection. And we introduced white blood cells to the blood channel. We then saw the white blood cells migrate across the capillary cell layer, through the pores of the central membrane, and into the airspace where they engulfed the bacteria.
Like a computer game.
Here's a video that shows this response in real time viewed through the device. The tissue cells are not visible here, but we can see white blood cells flowing freely in the capillary channel of the device, just as they do in blood vessels of a healthy person. But when we infect the air channel by adding bacteria, the immune cells abruptly stick to the surfaces of the capillary cells on the opposite side of the membrane, located directly below the infection site. Here's a magnified view showing a migrating white blood cell, making its way through the first capillary cell layer, wriggling through the pentagonal hole in the flexible membrane, and then moving out of focus to the other side. When viewed from the air channel, with all cells visible, you can see a round white blood cell popping up from below. Just like in a real lung infection, the white blood cells, which are now colored red, engulf and kill bacterial invaders. The lung on a chip can also mimic the toxic effects of airborne nanoparticles on the lung. In fact, Experiments with the device led to the prediction that breathing increases their absorption into the bloodstream, and this has been confirmed in animal studies. Bio-inspired microdevices that can mimic whole human organ function, such as the lung on a chip, could potentially replace animal testing and bring new therapies to patients faster and at lower cost in the future.
So I thought that animation very nicely shows the high level of systems biology that it can execute with the help of microfluidic devices. Using a very similar device, you can also do a gut on a chip. So here we've got the typical kind of crypts that are available in our bowel cells. So our bowels are all developed to absorb nutrients, which means that our bowel cells create a very high surface to volume ratio. The way that we do increase our channel length and increase mixing in microfluidics is using the zigzag channels. Here, similar geometries have been developed to create the crypts where bowel cells absorb the nutrients from the food that we eat. And these crypts, there's also all villi in order to aid the mixing and not be reliant on diffusion only. So the same strain, because the bowels work by peristalsis as well, so the same kind of strain and relaxing that was relevant to the lung cells is as relevant to the bowel cells. And successful formation of this villi and as well as the crypts that allow for the nutrient exchange have been demonstrated using organ-on-a-chip devices with these side channels as well. The second type of microfluidic devices that are good for culturing organ-on-a-chip devices is ones that can have a gel of extracellular matrix hold in place, but with some guiding structures patterned on the bottom of the fluidic structure to allow two different flows of one or two different cell types and study their migration into the extracellular matrix gel that contains all the nutrients that they're happy with. So we can have flow channels here where epithelial cells will grow on the wall. Other cells can migrate into the extracellular matrix and potentially meet and greet. And this is one of the systems that has been commercialized a couple of years ago by MIME-TES and has been readily adopted by the pharmaceutical industry. So here we have a movie how this the MIME-TES system works. Sound from your way. Shouldn't be clicking it. So it's all done in the microtypeplate format that all the cell biologists are used for to work with. And then each of the reservoirs in this 96 well plate has got its own fluidic structures that allow you for controlled co-culturing of different cell types. Because it's a fairly viscous liquid, it doesn't spill over the edges. Load the different cells. You can see it's a hydrophilic substrate, so the aqueous liquids wet the surfaces, you load the cells. Pink stuff is always the media. Second cell type. Tilt, we can make sure that we've got a flow and we don't need to pump, we can just use siphoning because the devices are being dropped back and forth. And then we can get structures like blood vessels being formed. Look at the formation of new vessels. different formats that are available to conduct different assays. That way it doesn't drop anymore. To conduct different assays for organ-on-a-chip devices, using all methods that, or all techniques that the people trained to work in a molecular biology lab are already used to work with. So this has all been developed with the end user in mind. Pipetting, using multi-channel pipettes, using plate readers, that's all common technology for anyone working in a biology lab, which means that this technology is very accessible for the manual handling of the technical personnel in the laboratories, and they actually don't need to know anything about microfluidics, laminar flow. They just are instructed which reservoir, what solution or cell suspension needs to be loaded in, and the assays will conduct themselves. In addition to having the hydrogel sitting in the middle, these devices create little cavities in a hydrogel by initially starting off with some glass mandrels, some glass tubes. After the hydrogel has been set, you can pull out these tubes because they don't, the gel doesn't stick to them, which then means that you've got cavities where you can start seeding cells in, and these cells will then, if they're epithelial cells, they will automatically self-assemble on the edges into vascular structure that you can then, for example, use for doing studies into the development of cardiovascular disease. This in here is another example, particularly looking at vascularization. And then we've got microfluidic channels that allow at a macro scale how we distribute our nutrients and seed some cells. But there's a large cavity here that originally at the starting point only contains the matrigel. However, cells like building connecting fluidic networks. And what happens if we've got the right nutrients present in this matrigel is that vascularization happens automatically, and that again gives you the microfluidic plumbing that allows you for the delivery of nutrients. And at the same time, you've got a biorealistic capillary network that has self-assembled and grown and developed by itself. So here we've got an example of that, where this is the little triangle filled with the extracellular matrix, and we've got the fluidic inlet and outlet channel sitting on the top and bottom of the screen.
Developing a successful cancer-fighting drug averages 10 to 12 years of testing and investment of over $2.5 first to lower the cost of drug development test that's focused, but its impact.
There we go.
Growing cancer cells in two-dimensional sheets, but human tumors are more complex, multicellular, three-dimensional structures, which alters their response to anti-cancer drugs. We grow cancers that retain all of the characteristics of human tumors. Our proprietary human vascularized microtumor platform, or VMT, contains three-dimensional microtumors embedded in a natural scaffold. Tumors, like all tissues, need nutrients to survive, so we've incorporated a natural network of blood vessels into the VMT so that nutrients can be delivered to the tumor in the same way they are in the body. Here, the tumor behavior more closely mirrors humans, leading to more accurate identification of effective drugs. Our platform is simple to use, yet sophisticated in design. The full system of 16 individual VMTs fits on a single palm-sized self-contained plate. The living capillary network in each microtumor connects with two outer channels and carries blood substitute that provides nutrients to the growing tumor, just as occurs in a patient's tumor. Multiple tumor types have been created, and when anti-cancer drugs are added to the blood substitute, its effect on tumor growth is readily observed and measured. In addition to tumors, the living blood vessel network can also support other tissues used in disease models. We call these vascularized microorgans, or VMOs. Today, these platforms are providing scientists powerful new tools to enhance the accuracy and speed of drug discovery and validation, as well as...
So the rest is again, please buy my product. But this is a really nice tool. Again, it is accessible by people studying human biology, studying tumor models, or even loading a little bit of a tumor biopsy of a specific patient to see what the likelihood is that this tumor will respond to a certain cytotoxic drug. We have some simpler plate systems as well, where we've got some fluidically linked trans-well plates, where we can culture epithelial and endothelial cells at either side of a membrane insert into a microtiter plate. But then these plates are also equipped with a fluidic network connecting the different cell culture devices, which means that if we can culture different cell types under their preferred conditions in the different reservoirs, but the signaling molecules that they produce are transported through the flow underneath from one cell type to another, allowing for the creation of a simulation of organ-to-organ interaction, just as you would have with the metabolism and the distribution of a drug in the human body. We have similar devices that allow you to look at airway infection, where we have parallel channels separated by a rigid membrane, and then one of them is exposed to air, where we've got a long epithelial cells, a membrane to hold them in place, and then a flow that can mimic the blood flow to look at the absorption of nutrients as well as the transfer of red blood cells from the bloodstream towards the source of infection. There are some ways that we can also introduce features into the extracellular matrix, where we then can remove that and fill it with cell culture systems to again create cylindrical channels where slowly with time cells will self-assemble on the surface, creating vascular structure of epithelial or endothelial cells. And a lot of the different organs in our body work with any kind of vascular structures. Here we've got another way of people that connected, fluidically connected transwell plates, where we can have a series of different organs sitting together, circulating a drug, and looking at the metabolism and the interaction between the different cell types. If we then look at how we can create these little spheroids, these little functional droplets of organs, we've got a couple of different ways. One of the ways that we can create these spherical particles is using the so-called suspended droplet method, where we've got a little capillary tube coming down, we've got a droplet hanging underneath, we can slowly top up the liquid that we are losing here by evaporation, and we've got cells sitting here, self-assembling on the bottom, and forming a spheroid-like culture, structure on the group, because cells don't like to float just by themselves, other than blood cells. Most of our organ-based cells, they like to grow together into a function. Once our spheroid has developed to the large enough size, we can expel the spheroid into a microtiter plate for further assays. In addition to that hanging drop method, hanging in the solution using magnetic interaction, we can just pipette cells of different populations in stirred microreactors and hope that they find each other and form clusters that way. And we can use droplet microfluidic approaches to inject different groups of cells into a droplet, and hopefully that they will join themselves and form an organoid. Once we've got these devices, of course, the people that are investigating human biology are really interested to know what is actually happening on there, which then means it is really attractive to start introducing sensing elements onto the chips. And we've got a whole range of different sensors that we can add. We can look at mechanical sensors, where we can calculate what the shear stress is, what the flow rate of the media. Can we stretch and get back to apply some cyclic strains? We can look at optical signals from deformation of the surfaces or interaction with gold electrodes. We can do more traditional chemical sensing, where we can measure the oxygen level using an oxygen sensor, either coulometric or electric. We can add dyes that are pH indicators and monitor the pH to see if there's any local changes in pH. We can measure the glucose level, other signal molecules, and if we work with neurons, we can also add little electrodes that pick up the potentials and the currents that are used for cell-to-cell communication. So there are numerous cell-on-a-chip applications. We have our primary stem cells, organoids, and primary cells derived from the patients. We have devices where we've got a single organ on a chip when we're looking at the interaction or the interaction of inflammatory agents in the bowel. We don't need to have a whole body, we don't need all organs, we just need to have a functional bowel on a chip. And we have the multi-organ devices that are really useful in studying how the different organs play together. What this then can be used is disease modeling. Particularly if we're talking about rare genetic disorders, we can get the cells from these patients and see how these cells behave differently to the normal cells that we know. We can do toxicity testing under a whole range of different conditions, also those that with normal human cells we wouldn't like to do, including lethal radiation levels, or see how cells and pharmacology would act differently when women are pregnant. We can compare drug responses between different patients, because maybe the amount of paracetamol that's good for me to stop my headache isn't the concentration that you need. And we can allow for the selection of selective drugs, which is already done, for example, in breast cancer, where there are certain breast cancers that are known to respond well to certain treatments. At that point, you're tested for the gene. Using organ-on-chip technology, you could actually see if the tumor that you've developed would respond or not to the drug that could be prescribed, which allows the physician with a much more informed decision-making around the best therapeutic strategy to prescribe. So there's a whole range of applications. We can do the standard drug screening in the drug development. We can make a big step towards personalized medicine, where we all get the right medication prescribed at the dose that is the best for us. We can get much more biorelevant drug screening. So perhaps out of the synthetic lab, we have discarded a lot of active reagents that we shouldn't have discarded. We can also start look at combinations of drugs and see if having...
Can you put aside your dupatta? It is touching the mic and
Yeah.
it's creating a problem.
Okay. Put it back on the shoulder and let's hope it doesn't fall. Thank you. And it is a very nice platform to start looking at infection monitoring or development of cancer cells, as we've seen in some of the models where we could use vascularized systems to study tumor behavior for an extended period of time. One of the examples I'll talk you through with is a gut-brain model where they combined a gut neuronal cells to look at the interaction between and the brain, particularly in Parkinson's disease. Because in Parkinson's disease, it is well known that the patients quite often have problems with their bowel and with their brain, and there seem to be some synergies there. So what they did here is have a co-culture device where in fluidically connected chambers, there were cultures of bowel cells as well as cultures of the brain cells, and different nutrients, different diets could be given to the bowel cells, and then you could look at the response in the brain cells. And this was done on Parkinson's patients of cells that were known to have all the characteristics of Parkinson's disease, as well as ones that had been genetically modified to be Parkinson's disease negative. What this research has shown is that there were significant links between the short-chain fatty acids. If short-chain fatty acids were be added to the bowel system in the Parkinson model, there were actually significant changes that could be seen in the cerebral microphysiological system, in giving the evidence that there is a link in Parkinson's disease between what's happening in the bowel and what's happening in the brain. It was found that a certain particular way of cell death, called ferroptosis, was uniquely increased when Parkinson's patients' cells were given short-chain fatty acids, which suggests that the defect in Parkinson's disease may actually be linked to lipid metabolism, rather than what was previously thought an immune reaction of the bowel cells following the interaction with the short-chain fatty acids. So it may not be an inflammatory response, it may be a result of the metabolism. And it was very interesting that despite the neuron cells that had not been exposed to the short-chain fatty acids, through the extracellular markers and the connections between the brain cells and the bowel cells, if the bowel cells were given the short chain fatty acids, a response in the neurons and the ferroptosis in the neurons was observed. So there was a direct link between the exposure of the bowel cells on the Parkinson's disease model to short chain fatty acids and the dying of the brain cells in the Parkinson's disease model. And there are no other platforms at the moment where you could study the system in so much detail. Because if you go to an animal model or even a patient that's given their acceptance, it is going to be impossible to only expose the bowel cells of a patient to short-chain fatty acids, because under such controlled conditions, any human being would die. But if you've got a cell culture model, you can actually really start tailoring your experimentation with your chemicals to exclude certain reasons, and that then allows you to understand the disease development and the links in the pathology in a lot better way. So we've got an advancement where at the moment we may be going from 2D cell culture to organoids to still animal models, but it is to be anticipated that animal models will be increasingly replaced by the organ-on-a-chip models, because they are more relevant to how drugs will be acting on us as humans. Because in most of the organ-on-a-chip, human cells are cultured and cultivated, which means that the response will be much more like what's happening if we take these drugs ourselves. So the learnings from the gut, liver and brain system, we've just been through those. So that was the conclusion slide. So we do expect an increased uptake of these organ-on-a-chip devices, and as you've seen, the pharmaceutical industry has jumped big time on board. There's a huge amount of money going in the organ-on-a-chip systems, and that has allowed the development of complementary organ-on-a-chip systems that are all interfacing with the microtiter plate format. So they're all compatible with all the pipetting robots and all the infrastructure as well as skill that is already available in the pharmaceutical industry. So it has reduced the barrier for adoption by thinking about the end users, thinking about all the instrumentation available, build on that skill, networks to a level of understanding, and with that more advanced drug screening or better understanding of the physiological systems. So that's what's happening. Very little to do with analytical chemistry, but I think it's still worthwhile knowing about as a scientist that actually very good alternatives to animal testing are under development and are being adopted by the pharmaceutical industry. Any questions on the cells and organs?
So this 10-day course was a beautiful journey, starting from the basics and to the another of his advancement.
Yeah, and the nice thing here is that there's so much commercial activity.
Yeah.
Because it's like AstraZeneca is using very few animal models. They've just all converted to the new technology.
Yeah.
So it was 10 years ago, it was new. The past 5 years, the adoption has been really, really big because the predictive values are better, which saves the pharmaceutical industry time, and time is money.
Similar things can be taken up for the agriculture models as well.
I'm sure you can make a other on a chip and study mastitis. I know that there have been little breast cell organ on a chip systems that produce milk have been made.
Ah.
So yeah, for the dairy industry that would be relevant. See what drugs do transfer into the milk which don't. So, then we only have one lecture left, but I think we take a little bit of a break and drink.
Water?
Oh, I've got my water bottle here. And one of the most useful parts, if you're new to the microfluidics field, is to start looking at the papers and see if you can understand how they've produced what they had. And once you go from a product to understanding how the things were made, there is a process in engineering that's called reverse engineering. And here this diagram illustrates it very nicely with an iron, where we can start off having an iron in our hands because we've used it for our clothing. We can then optically look at it and see what are the different components that it exists of. Both physically, we've got a handle and we've got a water reservoir if we also want to do some steaming. We've got a part where the cord needs to come out. But also functionally, if we want to have a steam function, it means that we need to have some kind of fluidic connectivity to allow our water to go into the heated element. Other important part, if we want to iron with high temperature, we probably need to have a heated element in there, which needs to be able to withstand high temperatures. We probably want the plate on the top of the iron to be homogeneously heated, because we don't want one area of our fabric to melt, whereas the other one isn't hot yet. So we can start thinking about how the 3D model looks like, create a mimic, and start assigning different functionalities, and then start understanding how this iron was actually manufactured and assembled. What were the different functional units that were in the device, and how have they come together? A cord, with the design of a iron, you probably didn't need to make a new cord. You could just interface the electronics with an existing cord that you could source somewhere. Rest of the layout, the upsides could probably very well be made by injection molding because it's plastic. But the bottom here is metal. If you make that to plastic, it probably would melt when you were operating the machine. So if you look at any object around it, you can actually look at it and kind of start thinking, how did they make this? What were the different thinking steps? And move backwards from having a functional object in your hand to how would this have looked like on the drawing board? And if you do that with a couple of microfluidic papers this is the thing that they ended up with. What were the thinking steps? What were the reasons? What were the decisions made? And were these reasons, were they trivial? Or were they very important for the end product? Like the decision here to have a water reservoir is probably very important for the function, because you can't steam if you don't have a water reservoir. If this water reservoir is blue or pink, it's trivial, because it doesn't matter what the color is. If it's transparent, it's probably quite useful. If you're using your iron, you can actually see if there's still water in there or not. So transparent as a functionality is useful. Color, just trivial, and pick whatever the color is that suits with your brand or that you think that your consumer audience will interact with best. But if you start understanding what are the functional decisions that allowed for functionality, and what were the decisions made that were just because it was available, then you also understand, if you want to replicate that research, what are the parts that you don't want to change and what are the parts that you can actually have some flexibility. Did the people here use paper microfluidics because it was the best thing to do? Or did they use it because they had nothing else available? And should I be looking at other materials? With paper microfluidics, maybe they have used nitrocellulose instead of normal Whatman paper. If they've done so, they probably wrote somewhere in the paper why that was. Maybe it was the wicking rate, maybe it is because there are smaller pore sizes available and they want to have a very slow rate. If there's no information given, but they seem to have made a really odd decision, don't feel bad sending an email to the corresponding author and just see, all people doing this use Whatman paper and you use nitrocellulose. Was there a reason to? And then the answer could be, we run out of filter paper. We started a student with nitrocellulose and there we finished it. Or they may have done all the work on Whatman paper. It didn't work or it had very poor binding capacity. They then moved to nitrocellulose and everything worked beautifully. So material choice, there typically behind decisions, if there's an important story, it should be in a paper, but it is not always spelled out. But if you understand that the Whatman paper is not the same as a nitrocellulose membrane, and you know that all people that have done immune assays have used Whatman paper, and this one recent article that you found has used nitrocellulose, it may be worth asking the question, why have they changed the material compared to what the rest of the field has already done? Similarly, if people have used a laser cut fluidic structure from tape or an injection molded device in PMMA. The reason for picking one or the other may very well the availability in the lab. It is very easy to get a xerography machine and cut some double-sided tape to size as spacers. Whereas injection molding equipment, it's expensive equipment and it needs a sophisticated operator to run it. So not every university has got access to an injection molder. Commercial devices are quite expensive. Xerography using double-sided tape is really cheap. So the decision there may not have been made on the materials because they may use PMMA still as a top and a backing. The decision there was made, I can quickly prototype and get my student through for very little money if we use a laser cutter. If we ever want to upscale, we can think about injection molding, but at this stage, not yet. So understanding in a paper what has been used and trying to understand why certain decisions were made. There are some like-for-like decisions, and you can easily swap if you don't have a xerographer machine, so you can't cut, but you've got a friend in the engineering department that's got an injection molder. Maybe actually you can do a very similar experiment using your engineering friend's injection molder. Doesn't change the device and the geometry at all. Hi. And it is make the microfabrication do whatever you want to do. So if you analyze a paper in the literature, ask yourself the question, what was actually done? What was new? What was effective that was done? Do they argue how it was optimized, why it was effective? What does the author themselves say about what the novelty was and what the reason was that they did it? Because typically it's hidden somewhere in the discussion of the results. We use PMMA because PDMS is a sorbent and our analyte disappeared. Then you know that if you look for that paper for the same analyte, perhaps PDMS is not the one to go. Whereas if they just say they worked with PDMS and there was no problem and the analytical results look consistent, then probably the analyte that they worked with was not very hydrophobic and the absorption of the analyte into PDMS was not a problem. At that point you can make the decision, do I want to stick with PDMS, have a risk that there's absorption, but have soft lithography as a very fast and effective microfabrication method? Or do I want to look to see if I can get another microfabrication approach to work with a less sorptive material to not even have the risk that my analyte will be disappearing? The other thing to ask is, we have solved a couple of different issues here, but we may not have addressed all here. What are the issues that are still remaining? What are the challenges that the authors may have mentioned? Or what are the differences that I can see that the authors haven't mentioned at all? And does that provide me with an opportunity to do a new research project or to do it slightly differently? Every paper has got a different scope. Sometimes it's an assay that has been developed for a target that has never been used before. Sometimes a new functional module has been developed, a new type of light scattering detector, use of a multicolor LED to do multicolor fluorescence detection. Sometimes it's technology, which could be in the manufacturing, introducing the use of 3D printing for making these type of devices. Sometimes in the technology in the fluidic dynamics, introducing a new type of mixer to get better, smaller, more monodispersed nanoparticles being formed. Sometimes it is in putting everything together into a full system. Sometimes different individual components have been developed, either by the same research group or by different research groups, and someone is putting it all together into a single box. As we've seen yesterday, the development of a sample enhancer route system that does sample preparation, sample pre-concentration, the assay and the detection, doing that all in a single integrated platform in a single way is not an easy thing to do. So maybe the novelty of the paper is having sample preparation or sampling sample preparation, assay and detection, having it all happening in one box, because for this analyte and or this matrix, that hasn't been done before. Maybe it's a miniaturized total analysis system that's got an automated sampling system that can allow for it to last any longer. So it is important to understand what the problem is that the paper addresses and to understand if it's got an application-based focus or technology focus, because you can't really win on both sides at a time, because that would be like doing an experiment where you change the two parameters at a time and you can never figure out why it worked or most often didn't work, because you haven't isolated a single parameter, so you can't draw any conclusions on a single parameter. So papers will always have either a focus on the technology development, on the essay, or on the application. So it is important to start visualizing the detail. What does the device look like? How does it work? What happens if my sample comes in? How are reagents added? How are the different fluids manipulated? How was it made? Was it made by casting? Was it made by a replication technique? Was it made by 3D printing? And also get an idea of what the additional infrastructure is, because particularly if you want to repeat that particular experiment in your own lab. If it's using HPLC pumps, they require really high pressure. So if they use the HPLC pump, maybe this system is not going to be very effective if you don't have any pumps and you're relying on gravity feed or maybe a peristaltic pump. So every different pump that is available on the market has got a different flow rate range and pressure range. You need to understand that you can't, without any issues, swap one pump over from another. Because a pneumatic pump has got a really smooth flow profile coming out, because it's the pressure that just dictates the flow at a certain constant rate. Whereas if you've got a peristaltic pump, we've got these rollers. So we've got a little bit of water being pushed forward over and over again. So we will have a pulsed flow. Biological systems quite like that. So there's actually, in cell culture systems, there's no problem having pulsative flows, because our heart is a peristaltic pump. But if you talk about the chromatographic separation, maybe your efficiency will suffer if you try to do HPLC with a peristaltic pump. You won't be able to generate the back pressure, but even an open tubular system, the pulsing will deteriorate your separation. So again, the fluid mechanics are determined by the fluid control and the pumping system that you take. So understanding what pump they used and why, and quite typically it will be explained somehow a little bit in the experimental, is important. Was it important to do imaging? Do they show any imaging? Did they need a confocal system? Or would just be a USB microscope enough to do this? So what is the setup in the lab that you need in order to repeat the experiment that you would like to repeat in your laboratory? And it's always worth to just, were there alternatives? Would they have been equal, better, worse? And yeah, glass is better to do some organic synthesis, but it's also harder to make. So even though there is a better alternative, maybe there's other reasons like safety in the lab or cost that still led to the decision to work in a polymer device. Other questions to ask is, would I have done the same? If I would have that problem and I needed a microfluidic solution, would I have done the same? Could I do this? Could I do this differently? Could I do this better? And then by coming up with some of these answers, you may start formulating the way you're going to conduct that instrument in your environment where you are, with your collaborative network that you've got around, with the skills and the instrumentation that your supervisor already has got. So the big day tomorrow will be examination. And I selected 4 different papers. Professor Malik printed 2, so maybe we'll just do it on the 2 papers. And I picked them to be relevant to either nanoparticle synthesis or to agriculture. So we've got one paper that looks at a very fast mixer coupled with a very funky bit of detection electronics to study ultra-fast reaction kinetics. We've got an agricultural example here where there's paper microfluidic devices for the assessment of durian fruit ripeness. We've got a 3D vortex here for making core shell particles, and we've got the green synthesis of pepsin-doped gold nanoparticles. The ones that I've got available to look at here for you now are the durian fruits and the green synthesis of the pepsin-doped nanoparticles. So what I would like you...
Green synthesis?
The green synthesis I've got here as a printout, and the durian. So the bottom left and the top right corner. Otherwise I can send you the PDF if you want to have a look at any of the other papers.
Yeah, you can share.
but we've got the printouts here available. And what I'd like to do tomorrow with you, and you can come in groups, you can come individually, I don't really care, is to just have an informal conversation on seeing by reading those papers. If you can answer what the material was used to make the microfluidic device. What is the microfabrication approach that was followed? Was it soft lithography? Was it made in a clean room? Was it made by simply stacking some bits of plastic together? What microfluidic phenomena were used? Is it laminar flow? Was there a mixing element? Did they play around and did they use the high surface to volume ratio? Did they use wicking? So what of the microfluidic phenomena that we talked about in the first week did you actually see come back in this paper? And I'd like to know what you like best about the paper, and that is a question. Unless you don't answer it, you can't have it wrong. What is the kind of features in this paper that you really thought, that's clever, or I'd like to do that? So I'm not going to assess you on if you can repeat the Reynolds number. I'm not going to assess you on any of the physics. My goal here was to make you think how you in your type of research may be able to open the microfluidic toolbox and progress your own work. So, and if you've seen another microfluidic paper that's really close to what you do, I'm quite happy for you to bring that tomorrow as well. Don't get nervous. We don't need to have a 2-hour conversation because we only have an hour for all the interviews. I just want to have a quick chat to you to see if you could understand the paper, what you got out, because that also gives me a little bit of an insight how much of the information that I've tried to send to you, how much you actually received. And that then allows me to do better again next time. So feel free to pick one of the papers here, and if we need any others, I'm happy to share the PDFs.
Do you have the copies?
So these are photocopies of the 2 papers. I don't know where the other 2 are. Maybe they didn't end up in Professor Malik's inbox. But I've got all the 4 PDFs here, so I can just email individually, so you can read to them on the screen. But I thought they would be kind of relevant here to the audience.
Yeah.